Hey there.
Hey, how's it going?
Good. How are you?
I'm all right. Busy week.
It is. It seems like this week just exploded.
You're telling me. Uh,
He
this,
should be back on here any minute. Yeah.
no worries.
Have you met him before?
I worked with him in my undergrad. Yeah,
Okay.
so
That's what I was thinking. I know that he was your suggestion, and I felt like there was another connection. And then being in and off of the Zoom meetings today, I'm just like, okay, I feel like there's a lot of people that at least know him or know of him.
yeah, a lot of folks who work with like Brian Grenfell's group, like Katya, folks from her lab would probably know him as well.
That makes sense. Okay.
Hey, Diana.
Hey,
Hey.
hey, how are you? Thank you for inviting me.
Yeah, thanks for coming. I'm sorry we couldn't host you in person, but it's great to see you virtually.
Oh yeah, it's great. Yeah, thanks. Yeah, the meetings is good so far.
Great.
So what do you do with the recording? I just realized when I joined, it says recording in progress.
So I just used the recording for archival purposes. If you're okay with us sharing the recording, we do so on our YouTube. But if you're not comfortable with that, that's not a problem.
Let me think about it. It's just I feel like I present. I feel like I get like anxious when I know it's getting recorded.
No
Uh,
worries. We can just do it for archival purposes, and I can email you next week just to see what you think. Yeah, it's not that
okay,
big a
okay,
deal.
yeah, okay. So if it's good, then if I don't stumble, then we can share it. Not good, then I'll tell you no. Yeah, yeah. Okay,
Yeah.
thanks.
Yeah,
That's
this
yeah.
seminar is also part of a class too, so we like to try to at least be able to put it in for people who can't make it to be able to go in and view it.
Oh yeah, no problem, man, no problem. So
If you want
Madison,
to,
you're. Oh, go ahead. Yeah.
sorry, if you want to go ahead and share your screen just to make
Oh
sure
yeah,
that we're
let's do
able
that.
to take care of that.
Just, clear. Oh, wait a second. No, this is this thing where I always forget how to do it. How do you? There's a way to do it, right? Where you can share and then be in presenter view. Yeah. Here,
Usually
let me
it's like the
try it.
formation of two screens.
Yeah, you
It's
need
something
two.
along those lines. It's not mirror.
I think you can also like initialize the presentation as if like you're just going to show it through your laptop, and then you can choose to only share like the audience view screen. Um,
Yeah, that's right. I always struggle on. I know there's a trick, right? Let me just try again. So I think you have to. I think it's like you share and then you don't share, right, or something like this, or. Maybe let me just see if I can get this to work.
It's always such a process for an in-person seminar too. Every room at Emory basically has a different method for the screen sharing. So it's a great time.
Now you see that, right?
Yeah.
Oh, but now the only problem is if I do the laser pointer. Oh yeah, it's good. You see the laser pointer, right?
Yeah, it looks great.
Oh, okay, awesome. Yeah, cool.
So we've got people that are going to funnel in here probably just about now. So we're going to wait just a couple of minutes before we start. Then Madison will introduce you and we'll move into your talk.
Cool.
All right. Madison, you want to go ahead and get us started?
Sure. Hey everyone, thanks for joining the Zoom. Today I'm excited to introduce Dr. Shadi Sadroy, who's joining us virtually from the University of British Columbia, where he's an assistant professor in the Department of Mathematics and the Department of Microbiology and Immunology. I first met Dr. Sadroy when he was completing his PhD work at Princeton in the Quantitative and Computational Biology program, after which he moved to UC Berkeley's Miller Institute as a Miller Institute for Basic Research in Science as a Miller Research Fellow for his postdoctoral work. Um, Dr. Sadroy's work combines principles from the fields of immunology, epidemiology, behavioral sciences, ecology, and evolutionary biology to understand pathogen dynamics. And I'm really excited to hear what he has to share with us today, so I'll turn it over to him, but please join me in giving him a warm welcome to our IDAS seminar series.
Cool. Thanks a lot, Madison, for the great introduction. Very nice of you to say that. Uh, I don't think I combine too many disciplines. I just do what I think is interesting, and then I have to learn new things. Uh, yeah. So Todd had asked me to put in a slide of the background. So yeah, sorry for the wordy slide at the beginning, but I'm collecting affiliations. So I promise this is the most wordy slide there is. So I thought, yeah, I'll put a little bit of the journey so far. So I was an undergrad in Victoria, in Canada. I had an honors in combined math and stats, a minor in biology, and then I wanted to learn more biology, so I went and did a PhD, as Madison said, in quantitative and computational biology. And then I went to this like a great program, the Miller Institute at UC Berkeley, where I was a research fellow for 3 years, and now I'm at UBC, jointly appointed and equally in both departments, and also a member of other Biodiversity Research Center and Institute of Applied Math and so on. So yeah, I think it's if anyone has more questions on the journey part, feel free to email me. It's a little bit strange when and I think infectious disease dynamics is one where you can be between disciplines. And I think, yeah, there's a lot of excitement and enthusiasm, but also there's some challenges. So if anyone has any questions, please email me and we can have a chat. Okay, so the broad motivating questions that I'm interested in and that will hopefully we'll hit in this talk at least a little bit that I think relate to your program here is that how do susceptibility and immunity influence infectious disease dynamics? What's the interplay between evolutionary, immunological, and population level processes for infectious disease? Sorry, I just have this thing that's in my face. Oh, here, I'll hide the meeting control. Good. And then finally, how do social, and I think in humans, but as Yop and I were talking, it could be non-humans as well, but human behavior dynamics shape epidemiological trajectories and vice versa. So what's cross scale, right? So, I mean, I guess you guys all work across scales. So you don't need to see this, but I included anyways. So I've discovered I liked making these schematics. I felt like it was a nice pastime, and it also helps clarify my thinking. So this is the way I think of cross-scale dynamics. So it starts off with new strains that are emerging, right? And if you think of examples, COVID almost gave us an example for each of these levels, right? So emergence of new strains, where do new strains come from? This was a major question during the COVID-19 pandemic. And then if you go a little bit beyond the molecular, cellular and tissue transmission. So how do pathogens transmit within tissues, within a host? And that's to deal with tissue tropism. So there's a little lungs there that I put in, and that's basically highlighting this idea that, and it's true, it was probably thought to be true for Omicron, where Omicron might have been particularly successful because of its ability to change tissue tropism, right? So this is one of the reasons why that might have been successful pathogen, successful viral variant. And then if you think within hosts, so still within hosts, but now you have the dynamics, right? So you have viral load rising, and then eventually your anti-adaptive immunity kicks in and then controls the pathogen. You hope, anyways, right? And then you recover, and then the viral load goes away. So then, depending upon where you are along that trajectory, might change your transmission dynamics, right? And in particular, if you have prior immunity, you might have a different kind of infection. So here's a schematic. Oh, by the way, the colors will be the same throughout the talk. So once you recognize that this like darker red is a primary infection, then hopefully that stays the same. And then this primary infection can transmit along the immune spectrum, right? From fully susceptible to fully immune. And people that are fully immune, then they're not gonna get any transmissible infection. And the people that are fully susceptible probably get something that looks pretty similar to what they had before. And somewhere in between, there might be this pink, right, breakthrough infection here. And that might have different characteristics, different viral loads, different shedding. And then we don't know what happens after, right? So there's a little aside here. So this is basically that immunocompromised individuals might have prolonged carriage and might actually could be a source of new transmissible variants. So already we're feeding back to the first level, right? And then if we think, so now we're thinking about individuals and immunity, and if you have this pathogens are evolving and they can erode immunity through antigenic drift, right? So you have this strain transcending immunity, you're immune to everything after you've been infected, and eventually the pathogen changes enough that it can infect you. Alternatively, your own immunity can wane, right? So your antibodies or whatever other immune mechanisms that exist can wane and allowing reinfection by the original pathogen. And then if you have enough of these immune individuals, you can accumulate them and you get community immunity, right? So this is this classic concept in theoretical epidemiology. If you have R naught or R effective is less than one, you can invade. If it's greater than one, you can, right? This is still in a homogeneous population. We know the world is highly heterogeneous. And so you have regions that are tightly coupled, where you might have strong immigration, and you have regions where they're not strongly coupled and they might not get the new variants might not get seeded there. And then you have to worry about persistence and extinction, right? And new variants might persist in some places, and then we're back to this question of new variants, right? And so in the middle here, we have this individual behavior. And so this is a thing that a part that I added to the cartoon later, because I've become increasingly convinced that this can shape every layer, right? So whether or not you choose to interact with someone, whether or not you choose to wear a non-pharmaceutical intervention, or whether or not you choose to get vaccinated. Okay, so one of the major questions we saw with COVID, and I think this is more general in terms of pathogen ecology, but what are the dynamics of an emergent pathogen as it transitions to endemicity, right? So you have this susceptible population that's naive and you get this new pathogen, what happens? And there's lots of different aspects to this, but if you think initially in COVID, people were looking at this initial growth rate, right? Let's estimate the R naught, let's estimate how strongly it grows, which is super important. But then in the medium term, you got to consider other factors, right? And this, in particular, you have to consider immunity, the strength, the duration of immunity, and what about individuals that are getting vaccinated? What if you have vaccine hesitancy? And then how do those interplay with things like non-pharmaceutical interventions where we reduce transmission? What about seasonality? So we know that a lot of directly transmitted pathogens also exhibit seasonality. So how does that feed into all these dynamics? So we explored that in this paper in 2020 that I'll give you a brief overview of. and show you what we find. And this was in particular for SARS-CoV-2 in an era where we didn't really know what was going to happen, and we had very little data to inform us, right? So the basic modeling framework we use is basically we use this SIR bracket S model. That's a generalization of the SIRS model, and it's adapted from Morris et al. In a paper in Interface, which first presented this work. So basically, you have everyone knows the SIR model probably, or probably many of you know, but let's just cover it anyway. So you're susceptible, you're infected, you recover, and you have lifelong immunity, right? The SIRS model, you're susceptible, you're infected, you recover, and this is very transient. After a certain duration of time, you wane and you become fully susceptible again, right? In reality, we know that lots of pathogens, you don't really get completely susceptible again, but you also don't have lifelong immunity, right? Somewhere in the middle. So the beauty of this model is that you can actually interpolate between these two endpoints continuously. So we have the SP class. So these are primary susceptibles. They get infected into IP, primary infecteds. They recover, and then they're immune. And that complete immunity wanes into SS, partially susceptible individuals. And then they can get reinfected into secondary infections. And then you assume for closure that there's only 2 infections. Tertiary, quaternary behave the same. But we relax this in future work anyways. And then there are a couple parameters here that I'd like to draw your attention to. So there's this epsilon here. Epsilon is this relative susceptibility to reinfection. And then alpha is the relative transmissibility of a reinfection. So if you set epsilon and alpha to equal 1, then secondary susceptibles and secondary infections are indistinguishable from primary ones. So you can just add up SP and SS, IP and IS, and you basically get your SIRS model, right? And now if they're 0, and you also make doesn't really matter. You can either make the delta 0 or you can just add R and SS together. No one can ever become infected again. Then you end up back with your SIR model, right? So you have these 2 parameters now that you can leverage. Okay, now there's the birth and the death. That's just this is population turnover, right? Demographic birth and death. And then the beta is transmission rate, and then the gamma is the recovery rate, and then you have the delta, the rate of waning immunity. Okay, so this epsilon and alpha we don't know for COVID. Beta we can infer from related coronaviruses. So we can take a related HKU1 coronavirus at the time, right? And that gives us some seasonal cycle depending upon the latitude. And we tried different climates. I'll show you for New York City. And then gamma, you can also, people have estimated this. Back then, it was also estimated from early cases in China. Okay. And then there's the other part, which I told you about, the non-pharmaceutical intervention. So we'll just assume that this is going to be all taken care of in this beta term. So beta is going to consider seasonal cycles and also reductions in transmission for a fixed period of time. So the model equations are as follows for anyone who's following and who's interested. But basically, for the mathematicians, you have 5 dimensions, 5 equations. You can reduce them down to 4 because the population is constant. And then if you assume a neat little trick that the relative transmissibility is 1, and so you put all that characteristic into the susceptibility instead, which turns out numerically doesn't make much of a difference, then you can reduce it even further to 3 dimensions. Anyways, this is a technical point. And then what you're interested in is to see what happens in the medium term. This is what I'm motivated by. So we can look at these many scenarios, and here I pick 3. And they're a little busy, so I'll just walk you through what they are. But the bottom bar tells you when the non-pharmaceutical intervention's on. So this is dark, you're reducing transmission. Gray, you're not. So it's very crude. You have a long period here. Here you have a shorter period. And then the last plot here, you have a short period, then you relax, and then you're a little bit short period again, and then you relax again. The 3D plots here have time on this axis. Epsilon, so this relative susceptibility to reinfection. And remember, when you go towards 1, you're going towards very weak immunity, towards the SIRS framework, and towards 0, towards the SIR framework. Fraction of the population on this axis, and then solid and dashed lines, primary and secondary infections. And what you see here is that the first peak is unaffected by your value of epsilon. The color is just highlighting to visually increase the difference between the epsilon values. The first peak's unaffected. That makes sense. Everyone's susceptible. Immunity should have no effect. If you look at the second peak, you get this very interesting behavior where it looks like intermediate immunity maximizes the size of the second peak, right? Somewhere epsilon, so you get a bigger second peak as epsilon goes up, and then it reduces again. In the different scenario, so now we have a shorter period of non-pharmaceutical intervention, you get the opposite relationship. So what's going on here? Well, you have the seasonal cycle, right? So what happens is you have a seasonal cycle that pushes the peaks around, allowing for an accumulation of susceptibles, and then you get potentially delayed but more violent peak with stronger immunity. So this is kind of interesting and highlights that you need to know what the value of epsilon is to make any kind of prediction in the medium term. Otherwise, this is going to be very difficult, especially with all these moving parts. So what we were interested to do here is to look at synoptic landscapes, right? And back then, you might fault me, I don't have vaccines so far, and you might say, well, we need vaccines, but we didn't know what vaccines would look like. But anyways, we still included them in this paper eventually to see what would happen. So here we just assume very simply that susceptibles are get vaccinated at some rate, they gain immunity, and then they wane at a potentially different rate into the secondary pool. And here you can do some math, find the minimum vaccination rate required for eradication, which unfortunately we know that these vaccines don't block transmission very well. So this is not really attainable with our current iteration. But then we were really interested to look at what happens in the medium term with these vaccines. So here again, the same colors. Top row, you have no vaccination, and bottom row, you have vaccination. The left column is poor immunity, and the right column is robust immunity. Again, so it's pessimistic and optimistic assumption. If it's poor immunity, you also assume that the clinical cases, which are the top here, so this is severe cases on the y-axis, it's for 5 years. Some kind of secondary infections are severe as well. And the bottom panels each are area plots. So you split the population up into their makeup. And as there's constant population here, so the population, you can see how much of the fraction of each type as time goes through. And the key takeaway here is if you have robust immunity and a vaccine, you get elimination, right? But that's not the setting we're in. But if you had that, you would be good to go. In the poor immunity case, you get these very elevated cycles and of severe diseases as well, right? Immunity doesn't protect you to infection nor to severe disease. And then if you're in this case here with poor immunity and a vaccine, eventually the severe cases go down, but you still have infections, right? So this is probably more the setting we're in. Anyway, so you can basically show that the varying this epsilon parameter and the duration of immunity can have huge impacts in the medium term. But the initial wave, the initial peak looks very similar. Okay, so this is the simplest kind of model. And then there are lots of discussions, right? Then multiple vaccines were rolled out, not as one dose, but as multiple doses. And then there's a limit on dosing, on the number of doses. So then there's a big discussions on should we extend the space to maximize the doses or not. And there are a lot of verbal arguments. And it turns out we realized pretty quickly that we could just use our models to address these questions. So now we refine the model. And you can maybe disagree now that this might not be so simple anymore. The title does say simple models. Now there's, I think, quite a few more equations. But I think it's still simple in principle. You split the vaccines into 2 compartments, dose 1 and dose 2. Now each with their own secondary susceptible class, SS1 and SS2, each with their own epsilons, this relative susceptibility to reinfection, and then each with their own I class, right? IS1 and IS2. And then once recover, then you just assume that things start again, right? So they go into R, and then SS and IS. So I'll draw attention to, and then IV is the vaccine doesn't work, right? So you have individuals that get vaccinated, but they can still get infected. Okay, so the parameter of interest here is omega. So 1 over omega is the average time between the doses. And you somehow have to link that up to the dose administration rate, right? So this is new. So this is what we do here. So we have some cartoon here illustrating this relationship, right? So if you're at the optimal dosage, the optimal spacing, then I'm giving Yop a vaccine and I'm holding a dose back, right? And so then I get twice as many doses if I were to just vaccinate a 1-dose policy, which is why this is exactly half the value of the 1-dose policy as you increase the interdose period to infinity, right? You approach this asymptote of nu naught. Okay, so this is the epidemiological bit. We also consider evolution, which I'll describe in a second. And then you can look at what happens at the population level in the medium term. And here's the reference for those interested. And then we can make scenarios again. And now the scenarios have many more colors, many more types. It's a lot more complicated, and each plot becomes many more plots, right? But now let's just look at one. So here we have the scenario where we have less robust natural and vaccinal immunity and shorter and weaker immunity following one dose. And again, in retrospect, I can find you the scenario that I think probably fits best with what we experienced, but at the time we didn't know, and so we generate all of them, right? And we have the area plot again, plotted below here. The middle panel's cases in red and severe cases in black dash lines. And then some kind of schematic rollout for the proportion vaccinated. The columns are different dosing strategy. So 1-dose strategy, 24-week interdose, 12-week interdose, and then the recommended 2-dose strategy. Okay. What's interesting here is if you take a look at the 1-dose strategy, right? So basically vaccination initiates when this wave is going up. You manage to really snuff this initial wave down, right? And especially if you compare this to this 2-dose policy, right? But now as you look below, you have this buildup of orange individuals, waned 1-dose immunity. And I told you I'm looking at the case where there's shorter and weaker immunity following 1 dose. So then lo and behold, you get a really big second wave, right? And then a really big third wave. And you even get a fourth wave. And that's because you have these individuals, the waned 1-dose immunity, that are contributing to susceptibility very highly, right? In the 2-dose strategy, you have fewer cases in the medium term, but you paid the price by having a big first wave. So what's the solution? If you look at the 12-week or 24-week interdose period, you can actually see that you don't have this buildup of these susceptibles in the same way. So you're able to control that in some way. So the idea here is if you're in emergency settings, you do a 1-dose strategy, and then you can switch to eventually make sure that individuals come back for their vaccines. And you can do this quantitatively and look at different ratios. And anyways, I don't have time to present this in this talk, but you can go and take a look if that's interesting to you. Okay, so this is a bit more classic. And now what about this really high unknown of evolutionary outcomes? So what you can do is we can take the 0th order model and assume something about the phylogenetic curve. So the phylogenetic curve was introduced by Grenfell et al. In 2004 in a paper in Science. And basically it posits that you have this immune pressure at the x-axis, and the potential adaptation rate for a virus is a function of this immune pressure, right? And it has 2 key components: the strength of selection, which increases as immune pressure goes up, and viral abundance, which decreases as immune pressure goes up, right? So if you have no selection, you have no adaptation. And if you have no virus, you have no adaptation. So somewhere in the middle maximizes this, right? Okay, so now we can think of flu as sitting somewhere around the top of this curve, right? And now we're wondering where are these different phenotypes going to be? And there's 3 phenotypes in our model that can have prior immunity, right? Individuals that have been vaccinated and or immunized and who then get infected, right? After their immunity is waned. So those are the 3 kinds. And we look at 3 scenarios. The scenario 1, where there's little variation, they all don't contribute much to adaptation. Scenario 2, where there's really high potential with 1 dose and little potential with 2-dose or natural or infection-induced immunity. And then scenario 3, where all of them sit at the top, right? And then again, you could just project forward with different dosing strategy, look at this relative net rate of adaptation. And you can tell that depending upon the scenario you're in, the dosing strategy matters quite a bit, right? If you're in the scenario where there's very little evolutionary potential, then it doesn't really matter what dosing strategy you pick. If you're in either of the other 2 scenarios, the dosing strategy makes a big difference. So basically, you can use these models to try to infer something, to try to tell what you need to know in order to make decisions. So obviously, you can't go and we don't have time to make these tests, but we can at least bind the potential scenarios in the future, right? To generate a little bit more quantitative insight into this. Okay, so building on that, then there's lots more you can do with immune uncertainties. And I don't have time to cover all of it, but I'll just give you bullet points here. So if you're interested, you can talk to me about it after or email me or look at the papers. So we can also look at vaccine nationalism. So this is this idea where some regions hoard vaccines at the detriment of others. And we can show that actually sharing vaccines can have really important beneficial impacts epidemiologically and evolutionarily. We can also look at what happens. Remember, I told you we stop at 2 infections, but we can look at what happens if you have tertiary and quaternary infections, and what happens if you have different risks of long COVID at each type. And you can actually show that there's accumulating immunity can modulate the infection levels in the medium term, but you can still have elevated rates of long COVID, depending upon how clinical immunity protects against long COVID. And then I've also looked at severity blocking immunity. So what if there's two time scales of immunity here? So you can wane against transmission and then you can wane against severe disease. That also will, epsilon has a huge impact here for prediction as well. And then finally, with a student, when I was in Berkeley, we looked at this. What if you have a different kind of waning vaccinal immunity? And that might have important implications for pathogen invasion. So if you have a protected community, what's your vaccine? How much do you have to vaccinate? And what is that? How does that depend upon this shape of waning vaccinal immunity? Okay. So all these models have assumed that there's no mortality and that we're looking at the medium term. But what about long-term effects? So I mentioned a little bit about long COVID. Well, what if you have the really strong demographic effect, like mortality after recovery? So I got pretty interested in this a few years ago, and I started writing some models down for the epidemiological impacts of this, thinking I wanted to do more evolution, but I wanted to see what happens epidemiologically first. And it turns out that you can make a very simple model. You're susceptible, you're infected, and then you recover. So it's almost like the previous models, but now we don't even have an R class. And then you're potentially resusceptible to infection once you enter the secondary pool of susceptibles. And we can add mortality during infection, which is fairly typical. And what the new part that we can do here is add mortality after infection, right? And once we do that, we can write down the equations. And so this is the flow diagram, which you can write the equations for. And it's very similar to what I showed you before. Yeah, this is the reference for the paper in case people are interested in. And then you can do the analysis. And what you find out is that you can actually trigger sustained oscillations and on the very long term. So here's an illustration. So you have time on the x-axis and the fraction of the initial population on the y-axis. SP in gray dots, SS in dashed purple, and I in red. And you can see that what you end up with is you have these cycles on the order of decades. And so if you zoomed in, it'll be hard, the infections will be going up, it'll be hard to tell what's going on. But if you look out on the long time scale, you got these major population cycles. For the mathematicians here, what's going on is you have a pair of eigenvalues that cross this red line, and then you have a positive real part, and it's a complex eigenvalue, so you get a Hopf bifurcation and you get these sustained oscillations. Anyway, so if you don't have mortality after infection, this goes away. And you can see that here. So if this alpha S parameter, mortality after infection, is small enough, and by the way, this rate can be very, very small, and it's enough to trigger these sustained oscillations, you have a stable equilibrium, like you would expect. And then past this threshold, the cycle appears. And if you have disease-induced mortality during infection, you actually need even less mortality after infection. So together, they synergistically accentuate the cycling. By itself, disease-induced mortality during infection doesn't cause cycles, obviously. And then if you have robust immune response, so if you remove this epsilon parameter I told you before, you have it here as well. And if this decreases, eventually you stabilize the system. And this makes sense because these individuals are contributing less to susceptibility. If you examine a bit more into the distribution, into immunity and its role in shaping this, so then I got interested to see what happens if you actually add an immune period and what happens if you have a different distribution for this period. You can make this model anyways. You can just add multiple R classes, and this will change the distribution of time you're fully immune. And if you do this, basically you can prove that if you have one or two R classes and you have mortality during infection or after recovery, it's not destabilizing. If you're going back, if your susceptibility is the same as the initial susceptibility, right? But if you have post-infection mortality alpha SS, that can trigger, again, 3 switches in stability. So it's a bit different than what we saw before. So this distribution, they can synergistically complicate dynamics, right? A different distribution of immunity plus mortality after infection can make things rather complicated. And so this is what this model revealed. But then the next question that's obvious here is, when does this kind of pathogen trait evolve? So there's been lots of work done on evolution of virulence, including by many people at Emory. And then I was curious to see what happens, when can this alpha S evolve? And so you can model this potential invasion by a mutant that's capable of causing post-infection mortality in a very simple way. You assume that you have an endemic strain that doesn't cause it. So you have a pathogen that doesn't cause post-infection mortality. stabilizes. We know that there's an endemic equilibrium, and then you ask, when can you invade? By when can a pathogen that causes a little bit of post-infection mortality invade? And it turns out that once you do that, you can show that unlike classic virulence, post-infection mortality is neutral and not selected against in the absence of other trade-offs. And if you have a susceptibility mortality trade-off that can select against classic virulence, so mortality and I, but favor post-infection mortality. So this kind of shows us that potentially this trait, there's very little selection against this trait, and it certainly should promote this investigation further, right? What's constraining this trait evolutionarily, and why do we see it or why don't we see it? So together, I think so far, I've hopefully highlighted the importance of studying immunoepidemiology, and particularly across scales. We wrote this perspective in 2022, thinking about what do we need to answer these questions. And so in terms of humans, we think we would need large cohort studies where we look at individuals and keep track of their vaccination status, their viral loads and their sequences, and when they're infected, and their serology across time, right? So they're infected, they recover, their immunity wanes, they get reinfected. And if we can keep track of this, then we can really know what's going on. And we have to keep track not only the number of infections we have, but also tie up the infections with the immune life history. And then if we have household transmission data, both for primary infection and secondary infections, we can then probably estimate how much the transmission impacts of the immune status on the viral variant. So we can see how much evolution is changing the transmission rate. And then if we monitor across ages and degrees of immune competence, that might even help predictability more, right? Because a lot of this stuff is age structured probably, which I haven't really discussed too much, but happy to talk about it later. Okay, so now the big elephant in the room, I think still, which I promised you at the beginning, but we haven't gotten to yet, is this idea of social dynamics, human behavior, right? So how does this play into it? How does this play? So far, we assume that there's mass action, that there's potential for a homogeneous population, but in reality, we know that this isn't quite the case, right? And in particular, we know that individuals make decisions to wear masks, to get vaccinated, to change their behavior based on what they see and perceive around them. So there's lots of potential interactions with human behavior. Lots of people have done different work. So here's just a way that I like to think about how this works in the paper published online yesterday, actually, in Trends in Microbiology. So this is the way I think part of the way, like a small part of how individuals might shape individual decision making, right? So here you're deciding whether to adhere to a non-pharmaceutical intervention, deciding whether or not to get vaccinated, and what's affecting is the local peer behavior, right? So if I see a lot of my peers wearing masks or getting vaccinated, I might be more likely to do that. If I see that there's a lot of infections around, might also incentivize me to get vaccinated or to wear a mask, right? But also externally, right? So if I hear that all my friends in Princeton are getting infected, I might think that it's time for me to go get vaccinated. Likewise, if all my friends in Berkeley are wearing a mask, then maybe I should also wear a mask, even if I don't really know what's going on locally, right? But it's this idea, this external information is shaping the way I think. Policy can also affect me. And then obviously, once I adhere to some intervention, then I can reduce the transmission rate. I can have a change in my individual immune landscape. I can have a reduction in severe infections. And that might also feed back, right? Feeds towards it directly to the decision making, or even through shaping other decision makers and local infection levels, and potentially external infection levels can seed local infection levels and local behavior, right? So there's this, it's a large interconnected system, and we're here. So lots of people have done different bits. So I'll focus on what we've done in the last few years on this, and really focused on non-pharmaceutical interventions. So we begun wondering, when might there be a tension between individual costs and societal benefits, right? So when might an individual perceive it good for them to forego adhering to an intervention, but in reality, if everyone adhered, society would be better off, right? So we can do a simple framework. This is evolutionary game theory, so probably not the most familiar to everyone here, but basically the idea is you get some benefit. There's some benefit or some cost of interacting. So N is I'm not adhering, and I is I'm adhering. And this is the focal player. So this is me, and then this is the partner player, right? N and I. And if I don't adhere, and the partner doesn't adhere, then I get this payoff. you can view capital lambda as some kind of risk of infection. So it's both the risk that the circulating levels times the perceived risk you have of getting infected or from infection. And then alphas is this modulator of depending upon the strategy type. So if we both don't adhere, then we have this potential risk of getting infected. If I adhere but the partner doesn't, then the risk change. So alpha n is a bit different. And now I pay a cost, right? I pay a cost to adhere. It's uncomfortable or I have to pay money. The cost is could be psychological or actual monetary value. And if I don't adhere and the partner does, then I get potentially a benefit. I don't have to pay a cost. And if we both adhere, then we maybe decrease transmission even more. And then I have to pay a cost. So this is one intervention. And now remember here, the infection levels are assumed to be constant, right? It's all taken care of in this capital lambda term. Now we know that sometimes you can choose between interventions, right? You can choose to wear an N95 or you can choose to wear a surgical mask. They might have different costs, so the gammas might be different, but they might also have different benefits, right? So an N95 might be really good at protecting the individual and the surgical maybe less so. Okay, so this is the game. Again, we're assuming that the constant infection levels, and this is the reference. And then we can go and do some analysis. And what we find in the one strategy case, so the two player one strategy case, two strategy case, so one intervention, you can look at the intervention cost divided by the risk. So this is this x-axis. So this is basically the capital lambda divided by, or the gamma, the cost divided by the capital lambda. And we can look at the fraction of individuals that adhere, right, from 0 to 1. The black line is this line plotted. So you can see when there's high risk, everyone adheres, and that's the social optimum. Everyone should adhere. In the low risk, no one adheres, and that's what everyone should do, right? The society wouldn't be any better off. In this intermediate risk, you see something very interesting. You see this region where you have partial adherence right here. So some people adhere, but not everyone. But if everyone did, it'd be better off, right? Society can gain benefit from that. And the prisoner's dilemma, where no one adheres, but it would be much better if everyone did. So this is the region where interventions might, or where like some kind of top-down mandates or incentives might be particularly useful to increase this adherence, right? And then if what we were interested in is to look at what happens when you can choose between two interventions. And here we're looking at N95s and surgical masks, and we parameterize this with data from Bagheri et al. In a PNAS paper in 2020, looking at fluid dynamics through masks, right? So some masks are better, some are worse. And here we're looking at, again, now instead of one axis, intervention cost divided by the risk, you need two, right? You need the cost of the M mask and then the cost of the F mask divided by the risk. The left region plots what we see. So you see F mask only, M only, coexistence of U and M, U only, so no one masking, coexistence of U and F. So you have two kinds of masks, F and M, and U is unmasked. And then you have this bistable region. So it depends where you start, you end up in either case. What's interesting is you never see coexistence of F and M at the population level. So this is particularly intriguing. If you look at the social dilemma regions, you can see the gray scale bar illustrates the strength of the social dilemma. So where it might be particularly useful to incentivize mask wearing. The blue, there's no social dilemma. So basically you can use these models to get some insight into when interventions across scales might be beneficial. But something we're assuming here is that the infections are constant, right? And as someone who comes from disease dynamics, I felt like we need to include disease dynamics in these models because that could shape. There's some feedback between epidemiology and decision making that's important. And so we then moved forward and did this. And the way we did this was we now made the payoff dependent upon the force of infection. So beta I here, right? And C is the risk that you perceive of what the infection is going to do to you. So the risk from infection is the correct term. P is the benefit, the kind of benefit you get, right? So you adhere, then you reduce your force of transmission. And then if I pay a cost when I adhere and I don't pay a cost when I don't adhere, and if my partner and I both adhere, we get a slightly better benefit, and then I pay a cost. Then we use this, so now we can write down, again, the game theory model. The evolutionary game theory model gives us some imitation dynamics. So we're assuming some model for social learning like we did in the previous work. And now we can find out what proportion of population adheres, right? This is going to be between 0 and 1. And then we can look at the average effect of the non-pharmaceutical intervention on transmission. And that feeds directly into the transmission rate. So we have a coupled system here. And then we can write down the math and we can do some analysis. And what do we find? So this is the reference. What we find is that there's 3 potential endemic equilibria. And I'll walk you through what this looks like. So you have the transmission rate on this x-axis, the fraction susceptible or recovered on this y-axis, the fraction infectious on this y-axis. And note the scales are different so that you can actually see all 3 on the same plot. The top panel is a fraction of behavior. So purple is people that are adhering, and yellow is people that are not adhering. So as you increase the transmission rate, and here the transmission rate's basically equal to R naught because I assume that the duration of infection is 1. So as you increase the transmission rate, you have no infections. Eventually you cross the R naught equals 1 threshold, and now you have infections. You still have no behavior, right? No one's adhering. And then eventually, as you keep going up, more and more people eventually, now people start adhering, right? At this threshold here, boom, purple shoots up. There's adherence, and then eventually everyone's adhering. And so then you hop back onto this other equilibrium, right? So you start off with no adherence, disease, partial adherence, disease, and then complete adherence, disease, right? So there's 3 potential equilibrium. The interesting thing which you can actually glean from this, but you can actually show analytically, is that when you have this partial adherence, there's no dependence on the transmission rate. So you increase beta, the level of infection stay the same. So what's going on here? Beta goes up, more people adhere, which then reduces the potential benefit of the intervention, right? Of the increase in transmission. So you're basically canceling this out, and it just cancels out perfectly. So you end up with this increasing transmission having no effect on circulating infection levels. Likewise, if you decrease transmission, you have no effect on circulating infection levels, right? If you have partial adherence. So then this idea is, well, I'm wearing a mask in the grocery store, but now all of a sudden, if the grocery store says, oh, now we're restricting ourselves to 10 people, then maybe I don't feel like I need to wear my mask anymore, right? So there's this bottom-up intervention that I'm adhering to, that I'm making decisions that's compensating for any kind of gain that I'm getting from a top-down intervention. So a way to get around this is to incentivize people to keep doing the bottom-up intervention, right? So you have to think using a cross-scale approach when you're rolling out these kinds of interventions. You can also use vaccination. So if you have vaccination, you actually reduce the level of infections, and you'll see it right here. So this is for different values of P, the transmission rate on the x-axis and the fraction of infecteds on the y-axis. And you see the partial adherence level is still there, but as you increase vaccination, so the colors go up, you reduce this level, right? So vaccination's also beneficial in concert with this bottom-up intervention. Okay, so this is individuals' decision-making feedback onto disease dynamics. But then I'm going to close with something that's more of a hypothetical piece that I worked on with Wayne Getz that's now thinking about animal behavior. Oh yeah, I should actually tell you before I do that there's this commentary that Carl Bergstrom and Bill Handage wrote on this paper that really contextualized this piece. So if you're interested, I suggest you can take a look at this. I think they did it's a fabulous job, and they tied it up with some data as well. So it was really cool to see. Okay, so this piece on animal behavior. So here our idea is, can we leverage animal behavior to anticipate or predict potential zoonoses? So we wrote this commentary in Bioscience on this, and the idea that we had was to generate some kind of early warning signals approach using indicator species, basically trying to monitor passively animal behavior and movement and temperature through some kind of AI system to detect any change. And if we detect a change in some population, so we have to, the difficult part here is to identify the indicator species, right? So once you identify one, so this requires quite a bit of work, but if we could find an indicator species, we can monitor these populations, and we would know that maybe there's potential risk in one population, and we could go and sample. And this could be a way to try to anticipate it before spillover, right? Or right around the point of spillover. And that might actually be pretty important in a changing world. Okay, so to close now, I want to bring you back to the beginning where I had this big cycle, and I'll try to put what we've gone through so far. This I know is a cross scales talk, so I tried to highlight as much as I could across scales. So we've begun with the emergence of pathogens and their transition to endemicity. And I tried to motivate that the strength of immunity is a key parameter that governs this, and that there's a potential important interplay between immunity, non-pharmaceutical interventions, and seasonality. We then looked at post-infectious mortality and showed that this can trigger sustained oscillations and that it can actually readily evolve, right? And vaccine dosing regimes can also potentially influence evolutionary potential. I motivated these large cohort studies that are needed to study this interaction between immunoepidemiology and evolution. And then we went into decision making and social dynamics. And I showed that social dilemmas for interventions can emerge quite readily. And also surprisingly to me, that behavior can fully compensate changes in transmission in a simple SIR model. And then finally, we talked about using behavior and animal behavior to monitor the emergence of new strains. And then we're back at the beginning. So thanks to you for all listening, and thanks to all the collaborators and the funding sources and the various institutes and universities I've been at. And happy to take any questions.
I'll ask a question. So first off, this was a really, really interesting talk. And I will probably be looking at the recording of this later as well. Um, I'm wondering if or how well the findings that you got from theoretical models, if you've looked at like how they align with the behavior that we were actually seeing. And you did a lot of stuff during the COVID-19 pandemic and non-pharmaceutical interventions that are relevant for that. And so there's a lot of data there also from like empirically. So did you look at if the theoretical models that you created explain the outcomes and the behaviors that we were seeing during the pandemic well?
Yeah, that's a great question. So that's yes, I feel like so this trends in microbiology piece kind of calls for that. So I think so network epidemiology has done some like pretty fabulous integration. Starts off with conceptual models and builds towards this, I think really like a complete synthesis almost. I think maybe we need something similar in this decision making, individual decision making, these kinds of coupling. I mean, these models that I'm doing are really simple, right? So it's like we need to move beyond the conceptual model. Then we can start thinking about how to integrate data, I think. So I think that's a key point for the future, but we didn't do that. So I think I think it'll be interesting to follow this field. I mean, we had this, yeah. I think it's not my expertise either, but I think it's cool if people want to get going and try to figure out how we can make I think you probably even need something like I don't think it's complete imitation going on either, right? So there's probably, especially not in humans, so there's probably something else going on. Probably need more complicated models, but maybe you can eventually move past these conceptual insights into data collection. Yeah. So sorry, I haven't done the data bit, but so
No, it's
hopefully
okay.
we can learn
I
people
was
to do
just
it.
curious. I was just curious, like if you had kind of done a comparison. And of course, nothing against like simple models can sometimes be completely, they're good enough to explain the phenomena that we see. There's definitely a lot to be said for a parsimonious system that is adequate to explain some phenomena. So I'll be interested to see if anybody takes this and kind of compares it to just to see how well those map. But yeah, very interesting talk. Thank you.
Thank you.
Other questions? If not, I'll ask a question about your last part. Sorry, and you didn't really go into that. Just like the whole idea of, so within your One Health framework and then figuring out how can you detect any. So you're basically looking for anything that changes, like in terms of behavior. But like you said, the key is to figure out what species you're going to look at. So I wondered if you
Exactly.
can talk a little bit more about, what you would envision. Would you envision having cameras set up in caves where bats come together, or is this just like existing
I think that's an
things,
interesting question. We were thinking more probably. I mean, the bats one is kind of cool actually, but we were thinking more in terms of migratory birds, right? Like you can monitor birds, and I think there's some people that have started doing this kind of work, right? They monitor birds, see if their migration patterns change or something like this, right? And can you infer anything about the health of the population in that sense? I feel like it's a very non-technical piece that we wrote, just being like, oh, maybe we can bring in some of these. Maybe AI can be useful to us in that sense, really decrease the cost of monitoring somehow. Yeah.
Yeah, so I guess the thing is using existing, you know,
Yeah.
infrastructure, like they have those big radars and you can actually see
Exactly.
migration patterns as clouds on a radar.
Exactly.
So yeah, no, that's interesting. And of course, that's a really that I mean, it's also a very different level of behavior, right? That's really looking
Right.
at this population level behavior and
Right.
see how
Exactly.
that can then spur something going on here. That's cool. Well, if no one else is volunteering, I'll ask you another question. And we talked about this a little bit earlier, but just to get back to this post infection mortality, right? And so and we kind of talked a little bit about different behaviors too, that people that have become sick and are recovering and maybe it's not just how they so I guess the question is it just how they replenish a susceptible population, or is it also how they drive the individual behaviors of those susceptible population? I think you alluded that to that, but
Right.
you could
Right.
maybe talk
I
a
think
bit
it's
about
I think it's a good question. Yeah, I think so in this yeah, I think it would be interesting to explore, right? If there's so the finding emerged from this depletion of susceptibles due to mortality, but that's I think the strongest effect. Yeah, I don't know if like if you put in something else that varies, if you how strong you need that effect to be, like how much do you need to remove them? Yeah, I think that's interesting. Yeah, it would be cool to check. I don't have a good intuition quite now on the spot, but I think
it.
yeah, it'd be interesting to check. Yeah,
I'm sure there's millions of ways you can
yeah,
build on the cool stuff you have done. I think we all come up with
Thank
it's like, oh, if you change behavior this way or that way, or you died a little bit later and it affected your
right.
grandkids, how they're gonna mask and all that stuff.
Yeah,
It gets really complex, right? But it's
yeah.
interesting because it makes you think in a different way about, and that's what we were saying earlier too. It goes beyond this kind of network analysis, which was for a long time really the way if you think about human behaviors, but you're starting to look at the human behaviors in very different ways. So that's cool.
Oh, thanks.
Um, I have a question about SIR parenthesis S model. Like, how would you know the rate of like recovered individuals going back to susceptible, like
Uh
secondary? How would you
yeah,
know
yeah.
that?
So we don't know that. So this is a parameter we don't know. Yeah. And so we explore a range of those. Yeah, that's one of the ones you don't know. Yeah. So I think the nice thing with the initial, at least what I think is nice, maybe others can tell me if they don't think it's nice, but it's that you have only two. You can. It's so simple that you can pin down most of the other parameters except for that one and the epsilon parameter. And so you have two parameters you can vary, and then you can see what happens in the medium term based on those two different parameters. Yeah. So there's a Shiny application online too. You can go and play around with it. Yeah, I should have mentioned that actually. A lot of those immunoepi models for COVID has there's these online links. Madison has them, or she did at some point. So you can just go and vary the parameters, and then you can see what happens dynamically.
Yeah, I still have the links to those. I'm happy to share them with anyone who's interested.
So, and then maybe as a broad, fun question. So you've been assistant professor for a year now. And so if you could, and we always say, no one knows where they're going to be in 5 years, but what would you, maybe as a fun question, what would be your proudest finding that would get you tenure in your current position? Now, having done everything you have done, it's like, what's the one thing that you said, this would be amazing if I could pull it off?
It's a good question. I think like yeah, grappling more with these grappling more with the human behavior aspect, I think is pretty important. I think it'll only be increasingly relevant as we move forward, especially if we have another pandemic. And especially as people's behavior might be depend on, I guess, depending on how long the waning is, right? Because you still might, I think if we have another pandemic today, a lot of our behavior is going to be influenced by the previous, what we experienced during COVID, right? So,
Yeah, but yeah, in positive or negative ways, right?
yeah,
It
exactly.
would be
Either
really
way.
interesting, to see, if we're just gonna ignore everything because everyone was, ended up being so pissed off with all the
Yeah,
advice.
yeah, yeah. Yeah, I mean, that probably depends, right? If you have a really deadly pandemic in like 20-year
Yeah.
olds, then I think maybe the answer is different, right?
Mm. Yeah.
Yeah. Like I think that's where the pathogen characteristics really do the traits of the pathogens, which I think is also unexplored. Like how do these different traits affect, I mean, okay, minus like transmission rate or something like this, but a lot of these other phenotypes that we don't really think about, but could actually shape behavior too.
Hmm.
Yeah.
Yeah.
Yeah, I
Yeah.
think that's good. And I think like moving past the acute burden is something else that I think is only going to grow. Like all these, it seems like every other month there's a new paper looking at, oh, now there's this other long-term effect of infection. So I think we're focusing on the peak, but maybe we need to focus past the
Hmm.
peak too. Yeah.
Yeah. Yeah, and long-term effects, not just of the disease, but also the vaccinations, right?
Yeah, like if you exactly, yeah. I think there's lots of different interesting interactions that we don't think about that I think will be good to think more. Yeah.
That's cool.
Yeah, but I just
Okay.
Oh, sorry, I can't hear you, Katja.
I said, yeah, but autism. I was just joking.
I think I'm having some problem with
No,
my
I think it's Katja's break.
No, it's not.
oh
You're breaking up. Sorry.
I'm fine.
oh oh sorry, I wasn't sure where it's coming from. I saw your green and I was like, oh, you're saying something I can't hear. I'm always intimidated when people like Katja and Yap are in the watching me give a talk. So yeah.
I'm intimidating with Katja watches mathematical talks, but not me. All right.
Cool. Well, thanks for everyone for coming. Yeah. And yeah,
Thank
feel free
you.
to follow up on email if anyone has a question. Like happy to chat too. So
Perfect. Madison, I'll let you have the last word.
Yeah. Yeah, thanks everyone for being here and thanks Chatty for joining us. This is a wonderful talk and I'm looking forward to chatting with you later. So
cool. Me too.
yeah, all right. Yeah,
Thanks.
thanks everyone.
All right, good talk. If you want to take a break, your next one-on-one is at 4:15.
Great. I'm not sharing the slides anymore. Oh, I am still sharing them. Okay, let me just get rid
Yep.
of that somehow. How do I got rid of my remote control. So there we go. There we go. All right, so I'll come back here then. Thanks for
Okay.
listening too.
Oh, no problem. Great talk.
All right, we'll see you in 15 minutes, right?
Correct.
Great.